Hello everyone, I'm Dr. Arpita De. The topic for today's discussion is Southern blotting. Southern blotting is an extremely important tool in the field of biotechnology. We are going to discuss the detailed procedure of Southern blotting, followed by its advantages or applications. So let's get started. Blotting is a technique for detecting DNA, RNA, or proteins. The blotting technique associated with DNA is called Southern blotting, that with RNA is called Northern blotting, and that with proteins is called Western blotting. So, it is a technique for detecting DNA, RNA, or proteins initially present in a complex mixture. The key to this method is hybridization. Now, we should know what is hybridization. It is the process of forming a double-stranded DNA molecule between a single-stranded DNA probe, a single-stranded DNA probe, and a single-stranded DNA. So, a single-stranded DNA probe binds with a single-stranded target DNA. This is the picture or stepwise procedure of Southern blotting, where we can see the genomic DNA, which is pretty huge, pretty big, is made into fragments using restriction endonucleases, and then those restriction fragments are subjected to These restriction fragments are then subjected to agarose gel electrophoresis. After that, it is blotted on a nitrocellulose or nylon filter or membrane. After that, probes are introduced. Thereafter, they are exposed to X-ray film. So, a Southern blot is a laboratory method used to detect specific DNA molecules from among many other DNA molecules. This technique was named after its inventor, Edward Southern, in the year 1975. It analyzes DNA by transferring it from a gel to a membrane and using a labeled probe to identify specific DNA fragments. The technique is used to determine the size of a DNA fragment from a complex mixture of genomic DNA, and it is also relatively quantitative and can be used to determine the number of copies of a segment present in a genome. Since it is helping us to determine the number of copies of a segment present in a genome, we can call it to be a relatively quantitative process because it gives us an idea about the quantity, the number. This is the procedure of Southern blotting. The DNA is first extracted from the cell, then it is subjected to restriction digestion. That means restriction fragments are obtained using restriction endonucleases. Then they are subjected to electrophoresis, then denaturation, followed by blotting, immobilization, hybridization. Hybridization means probe hybridization, and finally detection. Now coming to the process. DNA is extracted from the cell and digested by restriction enzymes into smaller fragments called restriction fragments. Restriction fragments are then subjected to agarose gel electrophoresis. Now, once the gel run is over, it is removed from the electrophoresis tank or electrophoresis apparatus and placed in a glass tray containing denaturation buffer. After electrophoresis, the gel is blotted on nitrocellulose or nylon filter or membrane. The membrane is then removed from the blotting structure and attached to a vacuum or regular oven at 80 degrees Celsius for 2 to 3 hours. The DNA strands on the membrane can be immobilized by exposing the membrane to ultraviolet radiation. During blotting, the DNA fragments will be transferred by capillary action from the gel to the membrane. If you look at this image, see the gel is placed here. Just above the gel, the nylon membrane or nitrocellulose membrane is placed, on which several Whatman filter papers are placed. On top of that, absorbent paper is placed, and right at the top, above everything, a weight is placed so that it creates a pressure, and this also ensures that the gel, the membrane, and the filter paper, they are not getting displaced. On top of this absorbent paper, the blotting tray lid is placed, and on the lid, the weight is placed. This outer boundary is what? The southern blotting tray that has got a plastic support on which the filter paper bridge is there. This is all done in the buffer, and on top of that you place your gel, on which the membrane is placed, above which the filter paper, then the absorbent paper, and finally you cover it with the southern blot tray lid on which the weight is placed. So during this time, what is happening? During blotting, the DNA fragments will be transferred by capillary action from the gel to the membrane. The membrane is exposed to the hybridization probe, which can either be a DNA fragment or an RNA segment with a specific sequence that detects the target DNA. So after this, the membrane is going to be exposed to probe. Now the probe will go and bind to the fragments of DNA where it finds its complementary sequences. Hybridization is followed by washing with a buffer to remove the probe that is bound non-specifically or remain unbound, so that only labeled probes remain bound to the target sequence. Now, once the hybridization is over, the process of hybridization is over, it is washed. It is washed to ensure that there is no trace of unbound probe left. There can remain some probe that are unbound. That means they do not bind to any DNA sequence, or if they bind non-specifically, those are all washed out, so that only labeled probes remain bound to the target sequence, and it becomes easy for visualization and analysis. Now, the hybridized regions on the membrane can be detected via autoradiography by placing the membrane in contact with a photographic film. The images indicate the position of the hybridized DNA molecules, which can be used to determine the length of the fragments by comparing them with the marker DNA molecules of known length. If you, in a gel, if you have the DNA ladder is what? You have one well where you put DNA molecules of known length. That is your reference. With that, you keep on comparing the fragments that you get after your process is over. And by comparing, you get to know the sizes of your fragments. This is how it comes. See, after you have your probe, you expose it to X-ray films for detection and analysis. So finally, we come to the applications of Southern blotting. It is widely used in gene discovery, gene mapping, evolution, and diagnostic studies. It enables the detection of particular DNA segment, and it can be used in personal identification via fingerprinting. Determination of molecular weights of restriction fragments, which helps in the analysis of such fragments. So these are some of the important applications of Southern blotting that has made Southern blotting an indispensable tool in the field of biotechnology. So I hope you have enjoyed learning Southern blotting, studying Southern blotting with me. Till then, happy learning and stay tuned for new videos. Thank you.